Hello friends, this is Vaibhav Changedia. Today we have to learn a new concept that is recombinant DNA technology, their basic principle and applications. After studying this concept, learner will able to acquire skills on the techniques of constructions of recombinant DNA, cloning vector, isolation of gene of interest, and learner will able to identify the problem associated with the productions of recombinant protein. The steps which is involved in the recombinant DNA technology are: the first steps or principle, generations of DNA fragment; second step, insertions of selected DNA into the vector; third step, introductions of recombinant vectors into the host; fourth step, multiplications and selections of clone containing recombinant molecule; and fifth step, expressions of gene to produce desired protein product. So in this technology, first step, that is the generation of the desired gene fragment or DNA fragment. So in this step, we can get the desired GNA fragment or DNA fragment from the two sources. First source, that is called as genomic library, and second one, that is called as a cDNA library. In the genomic library, this is a collections of a clone which are containing the transformant, that is a whole genome is there. So due to the big size of the genome or DNA, we have to cut down that DNA into the small fragment or gene. So if you have to broken down that big DNA molecule into the small DNA pieces or fragment, that is called as a gene, we require one enzyme that is called as a restriction endonuclease. So that restriction endonuclease, it will going to be cleave that DNA molecule at a particular or specific location or base sequence, and it will going to make a cut or cleavage at the particular sequence. So that's why that DNA, it will going to be converted into the small fragments of DNA or gene. So that gene or DNA fragment we have to be used for the cloning. And second source of the getting this desired DNA fragment or gene fragment, that is called as a cDNA library. So in the cDNA library, we have to perform the reverse transcriptions. Okay, reverse transcription. So normal process is that the DNA are converted into the RNA and RNA are converted into the protein. But in case of cDNA library, that RNA, it will going to be back converted into the DNA. So that DNA is become a cDNA with the help of enzyme reverse transcriptase. So these are the two sources from where we can get this desired gene fragment or DNA fragment for the cloning. Second step is that we had got the desired DNA fragment that we have to be cloned. So in the second step, that DNA fragment we have to be integrated into the vector, suitable vector at a particular sites. So here different types of vector are used for the cloning: bacterial vector, viral vectors, yeast vector, like this. So in that, most commonly used vector, that is called as a PBR322, is most commonly used vector. It is a bacterial vector which they use for the cloning purpose. So in that vector, different sites are there, and antibiotic resistant genes are, that is called as the markers, are present in that vector. So we have to integrate that gene fragment or desired DNA fragment into the suitable site. So for this purpose, we have to perform the cleavage. We have to cut that vector at a particular base sequence so that we can integrate that gene fragment into the vector. So for the cleavage of the vector, again we have to perform the enzymatic cleavage with the help of enzyme restriction endonuclease. It will going to be cleave that circular DNA molecule, that is a vector, at a particular location, and it allow the integrations of the DNA fragment or gene into the vector, that is shown in that diagram. So in this way, we have to perform the integrations of the desired gene fragment into the vector. Second thing is that here two type of vectors are used in the cloning or in this recombinant DNA technology. First vector that is called as a cloning vector, and second one that is called as the expression vector. Cloning vector is used if we are interested only in the productions of identical copies of that gene, and expression vectors are used in such a cases where we are interested in the productions of the desired protein from that gene. After that, third step is that is called as the introduction of the recombinant DNA, which was formed in the second step. So that recombinant DNA means what? These two DNA was came together. One that is called as a gene fragment, that is one DNA, and another one DNA that is called as a vector DNA itself. These two DNA are from different sources. So these two DNA, they will going to be combined with one another, and it will produce a one new DNA molecule that is called as a recombinant DNA molecule in the second step. In the third step, that recombinant DNA molecule we have to be introduced into the host cell. That step is called as a transformation. So in the host cells, different types of host cells are used: bacterial host cell, virus, yeast, etc. But in that all, bacterial host cell is a preferable for the cloning purpose. There are some reason behind that. Is that first, it is suitable for cloning, easy to clone, and second thing is that bacterial genetics is well known. Genetics of the genetical study of the bacteria was already performed. Genome of the bacterial cell, example you can take E. coli. Mostly in that bacterial cell, E. coli is used as a host cell. So genetics of the E. coli is well studied and it is suitable for transformations. So due to these two reasons, this E. coli bacterial cell is used as a host. But naturally, these E. coli cell or bacterial cell are impermeable to this recombinant DNA molecule due to their size. So if you have to permeate that recombinant DNA molecule into the host cell, we have to give the calcium chloride treatment to the host cells. So that's why we have to increase their competence. We have to increase the competence. That treatment is called as a competence. So if you are giving the calcium chloride treatment to the host cell, it will increase the uptake capacity of that host cell. That is called as a competence. So after calcium chloride treatment, that E. coli cell, it will uptake that recombinant DNA molecule into them. So that is called as a transformation process. That is the third steps. It is shown in that diagram. So a reflection spot during the step 3, that is transformation, why bacterial cell is a preferable host. So pause the video and just write down the answers. Answers is that, just I told, there are two reasons. The genetics of the E. coli or bacterial cell was studied in detail already, and second thing is suitable for cloning. Then step 4 is that after the transformation, that recombinant DNA, it will undergo multiplications and it will produce the number of identical copies. That is the step 4. In the step 4, the multiplication is occur within the host cell. Multiplication is occur within the host cell and it will produce the number of identical copies. That is called as a exponential multiplications of the that desired DNA fragment or gene. And fifth step is that the after multiplications of the desired gene into the host cells, that gene, it will undergo expressions and it will produce the desired protein product. So if our aim is that we have to produce the protein product from that gene, we have to perform the step number 5. And if our aim is that just we have to get the number of copies only, so we can perform up to step number 4. So in step number 5, we are getting the desired protein product from that gene because the production of the protein is encoded by the genes. One gene, it will encode the production of more than one protein because near about approximately 45,000 genes are present in the cell and number of proteins are present more than that gene. So one gene, it will control the production of more than one protein. So these are the 5 steps of recombinant DNA technology. So these are the 5 basic principles or steps of the recombinant DNA technology. After that, one point that is called the applications of recombinant DNA technology. So first application is that the manufacturing of proteins and some hormones. So this technology is helpful in the manufacturing of the therapeutic protein, means the protein which are used in the treatment of some disease, genetic disease mostly. So it is called as a therapeutic protein. So this, by this technology we can produce the different types of therapeutic protein and hormone. Second thing, this technology is also helpful in the diagnosis of molecular diseases, example cystic fibrosis, tay disease, etc. So we can diagnose these different types of molecular disease with the help of this technology. Next is also helpful in the prenatal diagnosis. The most important applications of this recombinant DNA technology is that gene therapy is also helpful in the gene therapy. Then also it is able to produce the number of vaccines. That is example is called as a DNA vaccines, which can be produced by using the principle of this recombinant DNA technology. Next, different types of commercial and pharmaceutical products are also produced. And next, this technology is also helpful in the DNA fingerprinting also, and some agricultural application. So this is all about the basic principles and applications of this recombinant DNA technology. Thank you.